From Microbe TV, this is Immune, episode number 60, recorded on September 21st, 2022. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Good morning. How are you?
Back in our usual places. The last time we were in your office there, huh? That was cool.
Well, I'm still in my office, but you are not in my office,
yes.
Except for virtually. But yeah.
Also joining us from Durham, North Carolina, Steph Langle.
Hey there. Hi, everyone. Happy to be here. I'm in my office in Durham. Things are good. It's beautiful blue skies. Looks good out there.
Weather is cooling off. The nights are getting cooler. It's so sad.
Oh,
I really like hot weather. I was in Little Rock. It was really hot down there. That was cool.
Yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone again. Vincent, you shouldn't look at the upcoming forecast if you're not liking colder weather, because today is our last hot day for a while.
Yep.
Well, we're in the Northeast, and winter's coming. It gets cold, and I know
you're bracing for it.
I really like the summer. I just like the hot weather. I'm a Mediterranean, as I've said before. So, today we have a little bit of a different episode for you. We're going to talk about teaching immunology, and we have two specialists who are going to join us and help talk about this from Minnesota State University, Moorhead, Sumali Pandey. Welcome to Immune.
Thank you. Happy to be here.
What kind of weather you have over there in Minnesota? Is it getting cool also?
It's chilly. It's 50 degrees Fahrenheit today. So we're getting that fall chill. It's sunny though. So just a little bit of chill in there.
And from Washington State University, Phil Mixter. Welcome to Immune.
Thank you. Great to be here for the Big 6-0 and enjoying a crisp, cool morning on the Washington-Idaho border. I'm in the eastern part of Washington State.
Wow. It's good to have some West Coast representation. We're usually more Midwest and East Coast. This is good.
Yeah, I would be PNW, Pacific Northwest is how I identify.
So you're on the Idaho border, right? You're not too far.
That's correct.
How far from Idaho? Can you just walk there?
A whopping 8 miles. You can bike to the University of Idaho from the Washington State campus.
Cool. All right. So as I said, we're going to talk about immunology education. But first, I have an announcement, an exciting announcement where you can help support our Microbe TV science programs. A company called Vaccinated.us is very excited about the spike protein of SARS-CoV-2, and they've made T-shirts with the spike on it, which are very beautiful. You can go to Vaccinated.us and check them out. And for the month of September, they have agreed to donate all of their profits on T-shirt sales to Microbe TV, which supports all of our podcasts, including Immune, of course. And I just bought 2 on the train this morning. So you go to Vaccinated.us. There's a link at the top. It's called Spike Shop, which is cool. And there you click it and you'll see all the Spike shirts, different styles and colors. And there are 3 different kinds of spike on the front.
Wow.
It's a 3-dimensional representation of spike. Very cool. You put in the cart what you want, and then you put the promo code Microbe TV into the promo code slot, and you hit it. You won't get a discount, but it tells them to send the profits to Microbe TV. And hopefully, and this is for the month of September, so hopefully we can raise some money to support our shows. And we really are appreciative of Vaccinated.us for doing this.
Yeah, we love this. It would be great if they grow over time and there's a shirt for every antigen in the currently approved vaccine. That would be so cool.
Well, Matt, who contacted me a while ago, and really nice of them, they love what we do. He said, we think spike is the molecule of the year, and billions of people have volunteered to take it, and it's really done a good job in getting us back to our normal lives. So they wanted to, celebrate it, and what better way than to support science shows. So I think you vaccinated.
That's
cool.
very cool. See, I wish they had done it a little while ago when I did my immunology class where I talked about different epitopes on an antigen.
Mm-hmm.
It could have been really useful.
You could point it. Yeah, exactly.
Yeah.
Wear it around campus.
I mean,
you
I
still
have to
do
admit
that.
that
I would, but
I have been slow in getting. I've also been traveling, so it's been slow to do this. So
great use of visuals and props for teaching immunology, which is relevant to what we're going to talk about today.
But the funny thing is, we were very critical of the spike-only vaccines early on. We said, should we put all our eggs in our spike basket? It turned out to be okay, but okay, it is the molecule of the year. I think that's cool. I would agree.
Yeah.
Anyway, thank you, vaccinated.us. All right, so back to immunology education. Before we chat about it, we'd love to hear a bit about your histories, where you're from, where you trained, and how you got to where you are. Let's start with you, Sumali.
All right. So I was trained for my doctoral studies. I was trained under Dr. Jane Shu at North Dakota State University. I worked with a mouse model of fungal asthma. And with that, I was getting trained in both microbiology and immunology. And then I did my postdoctoral training under Dr. Joyce Ohm, where I brought in some epigenetic info into my mouse model of fungal asthma. I got funding related to that to work on it. So when I started as a faculty member, I was teaching both microbiology and immunology. And that's a funny story because that's what got me interested in immunology education passionately. I was familiar with all of the resources on the microbiology side of things with ASM curriculum guidelines and concept inventory and learning frameworks on CourseSource. And on immunology side of things, it was just scanty, and it broke my heart that why are we so scanty on immunology side of things? I love immunology. And I remember at ASMQ 2019, I was at a session on concept inventories. I came out of that session and I met, just like that, I spoke to Amy Cheng, who was the director at ASM at that time, that why don't we have a concept inventory for immunology? And she said, just like that, do you want to make one? And I was taken aback for a minute, but then I gave it a thought. I started talking about it at ASMQ itself. And when I came back, I followed up on it. So I contacted a couple of educators and got in touch with Amy again, who connected me to Phil and Sam, who were at ASMQ and immunology educators. So that's how we got started.
Sumali, how did you get interested in immunology in the first place? And were you always interested in science?
I was always interested in science. My mom is a math teacher, high school math teacher, and she used to complain about it, that you study more of biology than math. I would love you to be on this side too. But health and medical was what I really liked since I can't remember when. I wanted to be on health and medical side of things always. I did my undergraduate training from India, actually, in pharmaceutical sciences. So in India, we don't have to have a bachelor's to get into professional pharmacy school. It's after high school. Wow. And as a result of that, I was very exposed to the pharmaceutical side of things, not only the pills, but also vaccines and things like that. So I like the research part of it more than just being a pharmacist. And that's how I said, no, I want to do more research. So I did my master's in pharmaceutical biotechnology. And then I came to the States for my doctoral training. And thankfully, I got a lab to work in where I was working with fungal asthma. So I've had asthma growing up throughout my growing up years. So this was passion and interest and circumstances that kind of aligned together.
And you said that you did your PhD at NC State, so North Carolina State?
No, North Dakota State University.
Oh, North Dakota State University. Okay, I see. And so then you mentioned you went to Minnesota after your PhD. Is that correct?
Yes. So we are in Moorhead, which is right at the border with North Dakota.
Got it.
And I started teaching when I was a doctoral student as a TA first, and then as an adjunct. And I subbed for my advisor who taught immunology for one semester. So that's how I got into teaching very early on in my doctoral training.
Oh, that's neat. And I was curious, about that transition, because it really does seem like teaching was your passion. And for you, it made great sense to go from the PhD to adjunct faculty to faculty without you didn't need a postdoc, because you could stick with what you were doing and continue teaching.
Yeah, and I was adjuncting while I was a postdoc too. So I continued both of them on the side.
Nice. Okay.
Oh, wow. And you said concept inventory, and I want to make sure we get a chance to go to Phil's background too. But can you just briefly define what that is? Because that is not a term that I was familiar with.
Okay. Concept inventory is an assessment tool which is validated. So we have concepts and we have assessment questions that are aligned with these concepts that specifically very targetedly test those questions and those concepts. And as a package, when given together as an assessment tool, you can gauge where your students stand in terms of those concepts. So it's really a very beneficial tool for educators because they can gauge where their students are at the beginning of the course, in the middle of the course, at the end of the course, conceptually.
Cindy, you could kind of think of it as an external standard. So a lot of these concept inventories are validated or affirmed by our societies or people external. You know, if you get that tunnel vision in your own class, you're not really sure where my students should be nationally or something like that. So this kind of creates a consensus list and a consensus set of questions that align with them. And a lot of people have taken a look at them and provided input. So it really represents kind of the community coming forward with this is what your students should know and maybe one way to assess their progress. Apologize for interrupting.
No, that's great. There are concept inventories out there for a lot of different sort of biological subdisciplines. I too first became familiar with all of this through the ASM resources, and which are fabulous. But you can actually find them for sort of all sorts of different biology courses, and they are so helpful in first designing a course.
Wow. So Sumali, can you talk to you and Phil, I want you to be able to do your intro, so maybe we can wait until after, but that seems daunting you're the one who came up with the concept inventory for immunology. And so maybe we'll talk a little bit later about how you went about that, how long that took you, et cetera. But I guess we could go to Phil first.
Yeah. So Phil, tell us your history.
Yeah, thank you. I actually grew up in the Midwest, just outside of Milwaukee, Wisconsin. So, Steph, if you hear that Midwestern in my voice, it's back there. It's oh yeah,
that's very Wisconsin right there.
Sorry, but I have an older sister who's also a scientist, and she's a couple 3 years older than I am. And growing up, she was the one who was coaching me on, no, you should take AP calculus in high school, not just regular math. And so a lot of my scientific development was sort of modeling her behaviors, but also having discussions about what she was doing. So when she was an undergraduate, she worked in a lab during the summer. And it turns out when I got to the end of my first year of undergrad, I was going to Oregon State in the Pacific Northwest, beginning my love affair with the Northwest. And I came back to Wisconsin, and she said, well, you should talk to the lab I worked in at the University of Wisconsin-Madison, which was a lab run by Fritz Bach, who's a pioneering transplant immunologist. And I didn't have a lot of skills, but that initial undergraduate immersion in research got me off the pre-med track, and really, passion was fueled in the discovery. I did something kind of that made sense to me, but no one else had done in the lab. During an experiment, there were some space to run some extra samples, and I put in the samples that I wanted to know about, and that turned out to be very informative, was reproducible and ended up being a figure in my first paper that I'm a middle author on, because I really didn't understand everything, but I did. That discovery just really fueled that curiosity and that passion and got me focused on a research career. After my undergraduate at Oregon State in Corvallis, Oregon, I was a little burnt out on school. I was not ready to commit to the long haul of graduate work. I thought that's where I would go, but I wanted to take a break. Nowadays, we talk about a gap year or something like that, but I was really in that space of wanting to stay in science, but not necessarily wanting to stay in school. So I got a job at a biotech company in Santa Monica, California, on the west side of Los Angeles. And it was a small company I was employee 35, and it grew. I was employee 35 of 250 by the time I left. So it was an exciting time to be in biotech. And I ended up choosing to go to grad school at UCLA School of Medicine. I basically took a different bus to school instead of, I stayed in my same living situation. My wife and I were settled there, and I worked with Mitchell Cronenberg on intestinal lymphocytes, the lymphocytes that decide whether something's a friend or foe. At the time, we didn't know anything about those regulatory T cells embedded in the lining of the gut. We know a lot more about them these days, and a lot of progress has been made recently. So I became interested in immune regulation, did a postdoc at the Vermont College of Medicine, going from tail to tip of the country, I guess. I had some family in the Northeast, and it was fun to be in Vermont for about 4 years. And then I moved to Washington State to take an assistant professorship, run a lab. And to make a long story short, I really discerned that running a lab wasn't aligned with my passion so much. The more I did the management part, the less I did the bench work and those discoveries that I was so passionate about. And I really enjoyed the relational work with students. When I landed, I was setting up a lab, and they handed me half of an immunology class with a senior colleague. And that part was as gratifying to me as trying to write a grant alone in my office. So for me, I really figured out that what I love to do is watch that light bulb go on in the head of a student and watch them get it. That's the thing that really provides me a lot of pleasure. I've been doing that for over 20 years. I got off the tenure track and into what we call a career track, where my focus is instruction, and I got assessed on my job performance based on teaching and involvement in other things. But as Sumali mentioned, I won't ramble on further, the interaction with teaching-focused colleagues in our professional societies, and those collaborations have been really invigorating for me and provided some of my scholarship a little later in my career.
I love this for our audience to be able to hear the very diverse career options there are for those studying immunology.
Yeah, absolutely. And I think the other thing is that, I've discerned that, do what you love, do what gets you out of bed in the morning with a skip in your step, or however that works. And for me, that wasn't, running a lab, doing compliance paperwork, and writing grants. It's that interaction with students, whether that's in person or in remote platforms.
I love what you said, management, right? We don't get trained to do management. We just want to do science. And I know a couple of weeks ago, my colleague Amy went to the FDA, and she was complaining to me. And my colleague said to her, "Amy, welcome to management." And it's hard because I never wanted to do management, only in the sense that you make science go forward. So it's tricky, right?
Right.
Yeah, that is exactly a big part of my decision to be at an undergraduate institution, as well.
Although, to be fair, when you teach, you're managing the class, right?
It's a different kind of management.
It's a different kind of management.
It's a different kind of management.
It's not like you make sure everything's ordered and nothing breaks and this and that and the other, but it's different, but still management. Yeah. That's cool. Now you know each other, Phil and Sumali, right? How did you know each other, from what?
Well, the American Society for Microbiology, ASM, has these annual meetings of educators it's an older sort of title, but the Committee on Undergraduate Education probably doesn't really apply anymore. It's not that restrictive, but I think Sumali and I met first at a meeting in the teens at some point, and then when Sumali had this insight to begin to work on resources, I was one of the people in this meeting who is focused on immunology education with a couple of others like Samantha Elliott, who she mentioned, Sam.
So the American Society for Microbiology, would they be the main professional society that is providing resources for instruction like you've talked about, or are there others?
There are absolutely others, and we should just acknowledge that for every one of those disciplines, whether it's biochemistry, chemistry, biology, molecular biology, and cell biology, those sorts of societies generally have a subcommittee on education. And again, the resources vary. I think what we found is, I'm pretty sure Sumali is a card-carrying member, like I am, of the American Association of Immunology, AAI, and I think I got that title right. And what we found was their focus was a lot more on graduate education or postgraduate resources, and we didn't see a lot of kind of basic, fundamental undergraduate resources. And that was where Sumali identified some deficits, some gaps that we really thought we could fill. Again, just applying some of the lessons learned in these other disciplines and understanding what people had done for a long time, and now inserting the discipline-specific knowledge of immunology, those immunology-specific concepts. Is that helpful, Vincent, in understanding that?
Yeah, for sure. Because I'm only familiar with ASM and ASV, which also has some education resources, but mostly for virology. And so I would have guessed that immunology societies would also do that as well.
Yeah, in fact, I think it was last week or so that there was a publication of some similar guidelines for in JV for virology.
Yeah, and again, getting those in the hands of educators to use and helping people understand you don't have to reinvent the wheel yourself independently, that your society supports you that way is really important, an important message for us today to get out to people. Look to find those if you're teaching any class, because they really come with a lot of support and a lot of scholarship behind them.
So let me just interject my experience here because it's kind of relevant. So I had a career as a research scientist, right? R01 driven research. But 15 years ago, I got interested in undergraduate education because we wrote a textbook. And so I started teaching at the undergraduate campus of Columbia. And I did it more and more, but it was not really appreciated at a medical school, right? Where my main job is to generate grant income. So I recently got a couple of awards for communicating, and none of my department acknowledged them, which I thought was funny. But, over the years, I did more and more teaching, and I always felt I wasn't doing what I was hired to do. And, you have that guilt feeling in the back of your head. So I think that my research career did improve my teaching. But now, just this summer, I closed my lab, and I'm just teaching, and I'm staying at the medical school. They're going to have to pay me to teach undergraduates. They probably don't like it, but that's what I'm doing. So there are other ways to go about doing this.
It's interesting because I'm also research R1 institution, tenure track, full professor, running research lab. But we do focus on teaching because I'm employed by the state of New York, being part of the Contract College of Veterinary Medicine. Part of my expectation of my position is to teach. And the primary teaching is in the veterinary curriculum. But then we also distribute funds around campus based on teaching undergraduates as well. So
it's very
interesting
different.
Exactly.
So at the Columbia Medical School, there's no teaching requirement. If anything, you teach a few classes to medical students, which is a whole different thing. But my doing an undergraduate class was very unusual. And then since then, more people have been doing it. And, the undergraduate campus wants to encourage it, but it's very difficult because it's not your main objective. So you have to really like teaching to do that.
Yeah. So where I am, it's usually around a 15% teaching appointment, but a lot of my colleagues on the other part of campus, the main part of campus, we say, because we're at the College of Veterinary Medicine, a professional school, they have 50-50 positions, and with a 9-month contract. So it's very different than medical school. So I just raised that because we're talking about different options for career that you could take. And so just knowing that those are out there, that one could choose that path. That where they do.
I was going to say, I like this conversation to be able to understand different positions have different proportions of effort or salary, AKA that goes into these different areas. And so Sumali, when you started, what was your teaching percent? Was it 100? Was it 80? What did it look like for you?
So ours is a primarily undergraduate institution, and we are assessed on 5 different criteria. So first is teaching effectiveness. That's the most important one. Second is scholarly activity or creativity. And that's where a lot of our scholarship goes. And I define it as research. It could be education-based research. It could be lab-based research. We do have labs for our biology faculty members. Cool. So I have a pretty active undergraduate research program as well. Nice. And expectation-wise, we have to show evaluations on all 5 of the categories. So scholarship, teaching, professional development, student development, and then university and community service. So there's not a split as such.
I see. Okay.
From the definition of the job, we have to have 12 contact hours, which is contractually designed. And but besides that, you can engage as much or as little in research, and I tend to engage a lot.
Yeah. That is just so neat to see how diverse the different positions are. And this position, Sumali, that you have reminds me of Brianne's position. Brianne, does this sound familiar to you?
Yeah, a lot of this sounds relatively similar to what I have. I always struggle when people ask me that question about percent that I spend teaching versus other things, because for me, it depends a little bit on things like which month of the year it is. Say in July, I'm mostly focused on my research, and am not teaching classes. Whereas in November, the classes have sort of taken over my time, and I'm spending less time with the research. I mean, you can see I have a research student behind me. So it's the same kind of thing. I'm balancing teaching. For me, it's 3 courses per semester with the research, and it can be either education research or it can be more specific lab-based.
Right. I think institutions have a lot of different configurations, and Steph, I really appreciate the opportunity to kind of talk about that, maybe break some of the mythology about what you have to do. If you're getting a PhD in a discipline like immunology now, a teaching-focused position is an option for you. I'll just say out loud that Washington State, my unit, is hiring right now for someone in biochemistry, so please apply. But we're looking for folks who want to focus on instruction, but maybe also have a lab sort of bench research or an educational research scholarship as what they do when they're not teaching. So that mix of more teaching and less research is kind of the opposite of Vincent's medical school model. And the other thing that's important to say here, and I don't want to keep us off topic too long, is that the incentives and the rewards are really important. So if you take a position where your appointment says you'll teach so much of the time, you should be assessed on your performance based on your teaching, not based on what someone, it's very difficult to do both at the same time. Cindy, we applaud you for juggling those things. I know that how difficult that can be. But the idea would be if you want to focus on something like teaching, to find a position where that's your job description and also your path to promotion. Nowadays at my institution in the non-tenure track, you can move up and be promoted to full professor as an educator or as someone who's doing scholarly work and teaching well, but maybe isn't bringing in a lot of extramural funding.
There was an article I read right when I started at my previous position in the Nature Jobs section, and it talked about the two types of positions as being like getting into the Winter Olympics versus the Summer Olympics. Two different sets of skills, but both sort of prestigious and cool things to do.
Yeah.
I had
Vincent,
go ahead.
Go. Vincent, I'll ask you a question, and maybe that's against the rules here. But
no rules.
When you pivoted, and I'm interested to hear about how the textbook sort of opened your view into, educating undergraduates and creating that kind of resource. But did you have to learn educational skills, because very few of us are trained in cognitive psychology or learning or education? We get a lot of specific training in the immune system or our virological systems. Did you have to gain skills that way?
No, I had to gain that. I didn't have any training. Just like all this podcasting, I never had any training. I had to learn. But I always tell people, scientists can learn stuff. That's not a problem. But I was like giving talks, right? So I was invited to give lectures once a year in an immunology course at the Morningside Columbia campus. And at one point, the professor said, you're a good teacher. You should teach a virology course. So I had just written the book, and I figured I could use the slides and develop a course. So I did it. And so that's 15 years ago. And I've learned, every year, because the class got bigger and bigger, and I made mistakes, and I had people who corrected them. And so I figure I learned everything on the job. I don't have any formal teaching training. My mother was an English teacher in high school, so maybe I got something of that just by osmosis. But when I hear about ASMQ and all the resources, I feel badly because I never learned from any of that. But I think you can do that. You can learn, and especially if you are willing to hear from people criticism and fix it, then you can really improve your situation. Yeah.
And Phil and I were discussing yesterday that during postdoc or PhD training, if upcoming students can get even, one certification or teach a class, it is so helpful, because anytime down the line, they can be, even if they're focused entirely on research, teaching can or will become very big focus right away as they join the faculty position.
That was something that was available to me as a postdoc. There was a place where we could have teaching sort of classes over lunch and get a little teaching certificate. And so that was incredibly useful, if only partially to understand the language of pedagogy and feel like I could actually know what I was doing when applying to teaching types of jobs.
Yeah, huge resume or CV builder by getting those certificates and showing that you're using some discretionary time to better yourself in professional development in that part of your teaching portfolio.
So I guess I didn't do that because I wasn't thinking about teaching when I was doing a postdoc, and I just wanted to run a lab, right? But then at some point I got interested, and to their credit, when I drew up the course plan and I applied to the curriculum committee at Columbia, they let me do it, even though I had not really done a whole course on my own. I'm not sure why, but they did, and from that point on, I learned, right? So that's another way you could do it, depending on where you are.
Right. Isn't there a quote where no one learns anything the first time you teach, the second time you learn a lot and they don't learn anything, and the third time you both learn? So there's something about working through the kinks to be able to get your course running in such a way. I'm not saying that you'll have to wait until the third time you teach the course for the students to learn anything.
And then it all changes when we shift to remote situations like the pandemic. I felt like we were retraining ourselves on so many different things.
Yeah. Well, and Sumali, so what was your experience like developing the materials for ASM, the course inventory? What was that process like? What did you learn? Can you speak to that?
Yeah. So, first, 2019, when we started out, we gathered as educators. And one of the things, I reached out to Dr. Tim Postian, who has developed microbiology concept inventory. And he mentioned the first thing that, well, you're thinking about assessment, but we don't know what concepts are important yet. So what about that? I said, well, that's our first step then. And we started talking amongst ourselves, 15 educators in the task force at that time, about, I remember writing my first draft of these are the 25 most important things in immunology that I consider important statement, and giving it out to everybody and invited criticism, and it got ripped apart and we wrote it all over again. So it went through a couple of revisions and everybody had an opinion, and we, came to a consensus what is truly important from that. That draft, after about 3 revisions in our group, went out to the community. So we sent it out to ASM, SABR, AI at that time, had their own thing going on. So this went on and we collected survey data from them. We revised it. We published a mini paper with that at that time, just gauging what is important for educators, immunology educators, what are they teaching right now and what are they not focusing on as much. And then after the surveys, we decided to have focus groups, which was through SABR. Justine and Adam, one of our task force members, they helped us organize the focus groups, but a lot of us, 6 of us, were involved in basically sitting together with the participants and discussing those concepts line by line and going over, wordsmithing. What is important? Why is it not important? And how can we phrase it in a way that it is conveying what we intend to convey? So focus groups was followed by another draft within our task force and surveys again and community surveys. So we revised it again. And then the last round was one-on-one interviews with about 10 experts, where we really just sat down and discussed the whole document, saying that these are the concepts. What did we miss? What do you think is not sounding the way we intended to? Are they aligned properly with the key concept, the overarching concept, and things like that? So there was a lot of conversation and the community really came together, and I would applaud that completely.
So I'm wondering how frequently that needs reassessment. Because one of the things that I was somewhat shocked about in one meeting I was in one time, we were talking about textbooks, and the people who are in math and physics who were there, they were like, why would you need a new textbook? We don't want to have our students have to buy new textbooks because it never changes. The only thing they do is change around the numbering of the questions at the back. So it's a new version, right? And so you just keep giving the same textbook. So we can save money for our students and just have them buy the textbook version that was from 15 years ago and it's fine. And I said, how can it, things that's constantly changing, right? There's new cell types, there's new information, there's not necessarily rediscovery, but there's rethinking about how processes that have been described for a long time, maybe not exactly what we thought they were, because now we have tools that reveal new things and so forth. And I'm like, our textbook is different every couple of years. And they said, well, how do you teach? Right? So part of that is, how do we figure out what the core concepts are, right? That's sort of what you're defining, right? And what are the extra things? Because sometimes the extra things are actually exciting because they're the current things you see in the news, like new types of vaccines that are having a huge public impact, or new cancer immunotherapies that are based on newer findings that are not part of the core concepts. So how do you balance the ever-changing newness and excitement of the discovery with what is necessary to be learning the language, learning the players, learning the core in order to be able to understand that?
So I would say, yeah,
go ahead, Sumali.
Go ahead.
I really try and bring those newsworthy items into class and use them as application or case models, and acknowledge that the students have to know some of that basic core information to understand this application. On the other hand, in some cases, it it's a discovery that breaks the dogma we've been working with for too long, or undermines some of the things we've already known. But that really models a great scientist who's constantly, answering questions only to ask more questions, and also acknowledges we don't know everything yet. I won't cast shade on math or anything like that, but there's plenty of room to do that critical work. We have more tools to fight cancers we couldn't fight, 5-10 years ago. And now we're getting the next generation of drugs. That's really exciting for students and motivates them in some ways to also circle back and start with, a CD4 positive helper T cell involved in regulating antibody production. So it's a great way to reinforce that core concept while just demonstrating that enthusiasm for a Nobel Prize winning discovery.
Yeah, so I would reiterate that going back to the core concept along with application is the critical part. So assessing in a way that your assessments are covering not only the cool stuff, but also the underlying key concept. For example, at the end of the day, a receptor is a receptor. A cell is a cell. Even though we have 50 different new cell types, if they know how a cell functions or how a basic receptor functions in biology, they can apply that to different settings. So that's why this concept approach that we adopted was very life science biology focused. And we stayed away from applications because I figured that applications if they understand the concept, they'll understand the applications. You have to make sure that, if you're bringing immunology, especially to introductory biology students who have not had basic biology background about DNA, mRNA, and protein, or how prokaryotic and eukaryotic cell, they need to understand those. And then on top of that, you add immunology context to it in a step-by-step manner.
Yeah. So I pulled up the document because I talked to Sumali about this and have looked at the immunology guidelines when I was working on my syllabus. And I would say that I find things like this to, in some ways, take away some of that anxiety, Cindy. Mm-hmm. So I'll just give you an example of something something that I can see in this. So there's a fundamental statement. The immune system is a coordinated network of macromolecules, cells, tissues, and or organs within an organism. And so each year we might learn about a new macromolecule or a new cell or a new tissue. Mm-hmm. But in the end, I can put that in as an example to help me talk about the fact that it is a coordinated network. And so in the end, sometimes I don't have to feel like my student is going to be at the end of the world if they don't know every single cell type that's in every single paper that you could find out there. If I don't manage to talk about all of those, it's okay. But if I'm talking about how it is in fact a coordinated network, and brings together all of these pieces, that's actually probably more important. And if I'm telling them how you take the info you learn in a new paper and fit it into that framework, that's more important than me actually making them memorize the list of all the cells. And so I find that so much less stressful in that, okay, I can tie everything back to that big picture statement. I don't have to feel like if I miss some little thing that my students are going to be having their world end. I don't know if that makes perfect sense, but that I look at things like this in that breadth that I think that you guys have done really well. Mm-hmm. So thank you so much, Sumali and Phil, as really helpful in terms of, I think about the stuff I might normally talk about in my class and how does it fit into each of those broader statements.
Thank you for saying that, Brian. So this is what we heard from a lot of educators, that, somebody having this document in place after community feedback, and I'm okay if I don't have my students memorize every CD marker on a cell or CCR4. What is that? My students don't know it, but they know what a receptor is. or what a chemokine is and things like that. So that's where I think community feedback really helped. It gives, especially for newer educators who are starting out immunology education and building their tenure promotion portfolio, this gives like a confidence check that, okay, I'm aligning with something that the whole community is coming together and putting together.
Sumali, where can someone find this document that we're talking about?
So email me. It is on the web, on CUBE's website, since we have used this for several surveys and interviews. It's out there. It's on CUBE's website right now. So CUBE's website, there's called Immunoreach. If you Google Immunoreach CUBE's, you'll find that website there. But you can also email me. So I'll email the most updated draft. And it's getting ready for publication. So it'll be out soon and in a lot of open air.
Okay, cool. And so when you say, I see CUBE's, QUBES. Got it. Okay.
That's great. An open source platform for a lot of groups. So you'll find a treasure trove of options there. So search for Immunoreach.
Oh, that's great. And I imagine that the resources and techniques needed for an undergraduate course compared to a professional graduate course can be different. And what's really cool is Sumali and Brianne are both at primarily undergraduate institutions. Cindy and Phil are both at veterinary colleges. And so I'm curious if you could speak to that. I mean, I can think about, okay, undergraduates are going to need more core concepts, and maybe we could assume veterinary students maybe have some of those, but maybe not. So could maybe Phil or Sumali speak to the differences that those two populations require?
Yeah, thanks for the question, Steph. And I'll encourage Cindy to respond and maybe Brianne and her experience. But I've had the pleasure of teaching professional students as well as undergraduates. Remember that many of the undergraduates who are choosing to take immunology it's not their last immunology course. They're moving on a professional track, which is going to lead to Cindy's veterinary course or something like that. For some reason, a lot of Washington State students, we have a big vet school. Cornell has a big vet school. Yeah. I think we trade students and faculty a lot. But what I like to tell students in my undergraduate course is this is very similar to the professional course I'm going to teach. One of the big differences is the timeline. The professional course is essentially a third of the time, a third of the contact hours, a third of the sessions. So it's highly accelerated. And the assumption is that if you're in professional school, you're a professional learner, that you need a little less management, a little less mentoring as you learn some of those things. I think my undergraduates are also, as Sumali mentioned just moments ago, making those connections from my cell biology class, my biochemistry class, my genetics class. And it's starting to create this tangled web of integrated interdisciplinary knowledge needed to understand that coordinated network. And so that's what I would say is the big difference for me. The pace is the thing I can tell you is definitely different. But the idea that as an instructor, you have to help them make those connections. Whereas I think in professional school, you assume they're making those connections to a degree. And that doesn't mean that professional students don't struggle with our terrible vocabulary. You know, immunologists make up 3 terms for something that's already named. And so that having to speak a whole nother language is part of the challenge. And with undergraduates, that's kind of how I frame it. Like these are terms that you're going to have to learn to speak the language. So when you talk to me about the immune system or you write a short answer response to an essay question, you sound like you have some expertise. I don't know, Cindy, do you have other insights? I don't know if you taught undergraduates or just at the professional level.
I've had the opportunity to teach undergraduate, graduate, and veterinary, and it's really interesting what the differences are. I don't know if the students at Cornell are different than at other institutions, but I feel like those that are choosing the undergraduate immunology, like you said so beautifully, is it's a stepping stone to something else. They're not just taking immunology because it looks like fun. Usually they're taking it on the plan to something else. It is fun, don't get me wrong, but it's not just like, let me pick an elective and stick it in my senior year. It's very purposeful. And so you can do things like just teach the basics, right? It's always nice to put things in context, but like if I teach them complement, they just learn complement. If I try to teach that to a vet student, they're just like, why do I need to know this? I don't care. What else is this basic stuff? And so I've discovered that when you're teaching at the professional level, it's so critically important to come up front with some sort of case or example or an animal or a person or disease or something where you leave a little curiosity at the end. Like this person showed or this animal showed up with this particular thing. And at the end, I'm going to tell you after you learn all this stuff, why that is, right? That keeps them engaged. And then they want to learn it. But it is harder, I think, to grasp those people when it seems so frustrating to me as an educator. It's like, you wanted to go to vet school. Why do you not want to learn this, right? What's going on? But they just want to know how it's going to apply to what they have gone to school for, what they're paying to go to school for, and what their end goal is. So you have to tell them why they need to know this. So that's really the difference. And then in the middle is, or on the end is the graduate education. Then they don't want to be spoon-fed information. They want to know what is the latest, greatest thing. They want to advance their knowledge. They really want you to throw things out that they can talk more deeply about, think more deeply about, and discuss rather than just be lectured at. So I think it's very, very different depending on which position you're at, what kind of education you're at. The only other thing I would say is my biggest struggle, and I'd love to hear your thoughts on this, is if you have a team taught course and maybe you don't have familiarity with this, how do you get the immunologists to be on the same page with the language they're using, right? This is a continuous tension of individuals that I work with. It's like, well, I grew up calling it CD80. I don't want to call it B71. And then the students think you're talking about two different things. And then I'm trying to meet with people and they're like, well, what are these two things? I'm like, they're the same things. How am I supposed to know that? And I said, well, it was in a figure in one of the slides of one of the 50 slides that you saw in one lecture of 7 lectures you heard that week. You might have picked that up. No, we can't expect that. So how do you try and make a consensus language so that students have less trouble? They're not trying to learn French and Spanish at the same time as learning immunology. They're learning one language so that they can apply that information and they're not being distracted by having to learn 7 names for the same thing. Do you have suggestions for that?
Well, I'll jump in here and no, I'm jumping again. Go ahead, Sumali, your turn.
No, go ahead. Cool. I'm listening.
I think this is a common problem with any team-taught course. And it's especially a problem if the team isn't there for the whole course, if people are just dropping in. And that is a common educational practice in a medical school where you're getting, the expert on this today, which is the joy of having that expert on your faculty. The problem is from a learner's perspective, from the student's perspective, it's not connected to anything. And it's just a one-off, session. So I would affirm you to try and meet with your team and acknowledge, I've heard this from students, and ideally the other team members would somehow review your sessions to make sure that they're not duplicating something, that kind of coordination is common, but that they're also using the same terminology, have a meeting and come up with some consensus terminology, publish a glossary if that's useful for your students of some key terms that we've decided you should know. Maybe that becomes a resource that they populate with definitions and annotate themselves to create their own learning resource. Sumali, I cut you off, so this is your chance now.
Okay. So again, same thing. We realized this, with our vision and change framework. Vision and change is for life sciences education. It's not for immunology or genetics. And the overarching concepts, structure, function, and information flow and things like that are pretty common across the board. So using that as a common language really helped us to build fundamental statements that can be understood by a geneticist as much as they can be understood by an immunologist. So one way is that, but that's at an undergraduate level. I know that going further at a professional school level, there is a whole world out there with immunology terms, and we just came across one, antigen and immunogen, in our textbooks, and there was so much disparity in defining those terms in textbooks, which is like, okay, if textbooks are like this, I can only imagine how our students, or going further, our public would feel about immunology-based knowledge. So I think having that updated conversation amongst educators and policymakers for this would be really good, that we can have some kind of consensus on basic terms so that, and revisit them again, revise them again.
I really like Phil's idea that he mentioned. One of my colleagues for one of her other classes has her students use the course website to make a course-specific kind of like little Wikipedia where they can look up different course terms. And I am sort of like, oh yeah, I could totally do that for the immunology vocabulary, because even with the students just having to deal with me, the vocabulary is hard enough. I think that's one of the reasons why I have always been inspired by the work that Vincent does with making recordings of his lectures. And that's why I have my lectures on YouTube. My students have come up with all sorts of innovative things they do, partially because they're like, oh yeah, I want to hear those words again. But now that you're saying that, the idea of having them as a group, maybe a Google Doc or a thing on the website where they can make a class glossary could be really useful. I
wonder if you could motivate or encourage that, in that, it's maybe points that, depending on how many terms you populate yourself, maybe it's a bonus situation.
People do stuff like that.
Oh, neat. Okay.
You're gamifying learning now, Steph. So that competition is good. Yeah. Medical students are competitive. So it turns out. Pre-medical, maybe.
Yeah. And Phil, I'm sorry. I think I didn't realize, I knew you were at the veterinary school, but I didn't realize that you do teach both undergrad and veterinary students. And this can happen in the same year. You're teaching both groups, or pending?
So Steph, for me at this moment, I'm really focused on undergraduates, but I have taught some professional classes. Part of my history that you didn't hear is when I arrived at Washington State, I was part of a regional medical program of 5 states in the Northwest called WWAMI, Washington, Wyoming, Alaska, Montana, Idaho, teaching human medical students. And the University of Idaho, 8 miles away, had some of those, and we had some of those. In the time I've been here, we formed our own medical school, and I'm now in the 100% in the vet program focused on undergraduates. So my teaching responsibilities have changed over the course of my time here.
Makes sense. Thank you.
I could comment that I've taught both in the same day. It's very confusing. The same content.
Wow.
And mostly the same slides, but not exactly the same, because there are different depths and different focuses. And so it's been very confusing. I would not recommend that.
I would like to hear what kinds of immunology courses you have taught. There's the 101, which I get, but beyond that, can you think of subspecialty courses that you've done or would be interesting?
Yeah, Sumali, why don't you take that, and then I'll follow.
I teach a standalone course in immunology, but I also teach a non-majors course called Introduction to Public Health, where I bring in a lot of immunology. I have a graduate level course called Principles of Infectious Disease. Again, if you're teaching infectious disease, immunology is one side of the story that you have to bring in. And this is a graduate level course for teachers, in high school and middle school teachers who are teaching AP biology. So I felt that immunology was one of their things that they really wanted to update themselves on and learn about. In non-majors, I've had, the vaccine course. So that's another one that is out there. So there are different levels that can be. And microbiology, of course, that incorporates immunology as well.
Yeah, in my current configuration, this term I'm teaching an immunology lecture, which is really for pre-professionals and a couple of graduate students who've never had a fundamental course in immunology, but are in a lab where they feel like they need that information. So that's a fourth year or senior level kind of course for students who've had some basic information. We have a pretty big track of microbiology majors who want to work in hospital diagnostic labs as clinical lab scientists. So I teach a companion lab for this lecture course, which is a combination of immunology and virology, and sort of also professional skills are kind of mixed in there for a number of reasons. But so when we teach that lab, we're hoping students understand how diagnostic tests work when immune reagents are involved. We just did blood typing yesterday in that lab, and helping students understand where those antigens are, what a transfusion reaction looks like, why it's problematic for a student or a patient rather getting a transfusion. So that's a big part of the kind of work that we do, specializing for people who are headed to different trajectories. Like Sumali, I teach a microbiology course in the spring that has a section devoted to the immune system, because we're talking about pathogenesis and kind of both sides of the coin, pathogens and immune reactions. And I'll just add that I teach a science communication course for students earlier in their career with some basic communication tenets, but all the examples are science. And not surprisingly, because I'm an immunologist, many of the examples are public outreach for vaccines and other public health things that involve immune concepts. Even though those students aren't learning immunology, they're getting communication examples that have these immune examples woven into that. And maybe that's a good segue to talk a little bit about Immunoreach, but that's one of my interests.
Yeah, so I'll give a little background on Immunoreach, which got funded by NSF last year as an incubator project. And it allowed us the aim of Immunoreach was really broadening the penetrance of immunology into basic biology. So you don't have to wait until your senior year to take upper level immunology class. So there are instructors who worked with us because of that collaboration, that are developing resources to bring in immunology into human anatomy and physiology course. For example, they talk about RAS, renin-angiotensin system. One of the common link is ACE2, which happens to be our spike protein target, and that kind of builds on the immune system side of things too. So bringing in that coordination of how these systems are interrelated and can talk to each other was something that's important to anatomy and physiology as well as immunologists. And I'll let Phil explain one of his examples that you've developed with Michelle.
Yeah, so Sumali put in a grant, and I want to acknowledge that Sumali anchored this project and really, with some other key individuals, created a situation where they put out a call for collaborators. And I, as a more senior immunology-focused card-carrying immunologist, maybe sought out someone I would never talk to otherwise. And that was a person who's 80 miles away at Spokane Falls Community College in Spokane, Washington, teaches a very general population of community college students who have very diverse interests. And our purpose was to collaborate and create a module for her to use an immune concept as an example that aligned completely with her plan and her curriculum and her program, where I brought the immunology expertise. She's not a trained immunologist, but I wanted, and with her, to really create something that would work for her students. We chose with a lot of meetings and collaborations. Collaboration here was really synergistic. I love doing this. And she sort of honed in on the tetanus vaccine, teaching her students a little more about tetanus as a disease, and then talking about tetanus toxoid, why you can't give people tetanus toxin at very high doses, and how the toxoid was not harmful. And to be honest, one of the criteria for focusing on tetanus is it's not very controversial, right? It's people don't argue about whether you should get a tetanus shot the way we seem to argue about whether you should get a COVID vaccine. And so it's also something most people have, again, and because of the nature of the disease, you can also argue that it's a very safe and effective way to prevent some horrific consequences. That module included, Michelle developed an online puzzle where you actually looked at the fitting of antibodies to different protein epitopes or those binding areas on an antigen. And students had to manipulate that online. And it also included a discussion section about their views on vaccination, what they'd heard, and using tetanus as kind of a safe place to talk about these concepts without stirring up some other information or misinformation they may have heard.
Wow. For two big reasons, I love that. One, to be able to collaborate with community colleges, considering, I mean, even in Raleigh-Durham, there's a huge opportunity for people to get specialized training and enter the workforce as technicians at some of these large, or, small or large pharmaceutical or bio companies. So being able to educate people in basic immunology is excellent. But then thinking about, I think you brought this up earlier, behavioral psychology, popular culture, how are people interacting with the news that they hear? What weird websites have they been on to skew their view of vaccines? And how do you remove those barriers? And so I love that the tetanus vaccine is a safe way to discuss immunological concepts without bringing in the baggage of COVID, for example, MMR. That's just really brilliant. I love that.
And from conceptual assessment point of view, it hits right upon structure function right there. So we are just using immunology as an example. That's just one way to integrate.
I was interested in the way you were saying that teacher was using the computer and computer modeling to do a puzzle. Can you also expand on any other resources that you've been able to use that are not your standard in the classroom, put up some slides and give a lecture? Can you talk about other ways that we can communicate and educate about immunology using perhaps technology or other methods?
Right. Well, we've touched on a couple of them, and I love we talked a lot about using cases and using, examples and putting a face on the disease that is an example of the concepts you're about to learn. So that, Cindy, you get a gold star in great educational design, because that's a classic way to engage your learners. We talk about active learning, something that is tactile like this, or even virtually tactile, allows students to really see, well, that doesn't fit together, even if it's just in 2 dimensions. So that can't be right, and it can't be a high affinity interaction when you use those sort of vocabulary terms that might be considered jargon. So I think a couple of things there, by working through a case, by trying to investigate an example. Personally, I send my students out to the internet because I know that that's where they're going to go anyway. They're going to Google things they don't know. And so we try and bring that skill of finding credible information in a sort of embedded way to evaluate, well, you found something that is published, but does the other information align with that? And I think that's a critical skill, as Steph alluded to today, in terms of dealing with the the flow of information that comes. You have to sift it. You can't just take everything on face value.
Right.
Is that helpful, Cindy? And I affirm some things you already mentioned, but getting students to do that work instead of and letting them discover rather than talking to them, asking rather than telling is a really critical approach that I think works well with immunology.
Yeah, I know.
Also props.
Oh, yeah.
Props.
Say more.
Wow.
So I have 3D printed versions of a few different things. So I actually have influenza with a couple of different surface proteins, and I have some mix and match heavy and light chains where you can actually see the heavy and light chain have to come together to bind to different epitopes and things like that. I've been teaching about antibodies lately, so my antibody comes to class all the time with me.
What's his name?
And so
have a name?
That's a good question.
Antibody?
I have not named the antibody.
Abby. Yeah.
So I try to use a lot of sort of hands-on models what the students can actually take a look at, in addition to trying to explain in other ways.
There are 3D printable models that educators can use.
Bunch.
They're not, if you have a 3D printer, it's the I don't know, the file that you would load, so it might not be as expensive as you think.
Yeah, for us, we have a couple of places on campus where they have 3D printers, and they're sort of interested in projects. And so I found some of those files and said, hey, I don't know how to make this, but it would be really useful for my class. And they think it's a cool project to work on. So they've 3D printed me all sorts of random immunology things.
One of you mentioned not delaying taking immunology till your senior year. Mm-hmm. And I think this is so important. So at Columbia, we have this core curriculum, right? First couple of years. And it has different units, it has a literature, it has a science unit, right? And the goal is to get students excited about science and also to teach students the role that science plays in society. Okay, so here are the 4 science units that are included. Mind and brain, okay, that's great. Biodiversity, that's great. Earth science, and then astrophysics. Forget astrophysics. There should be microbiology and immunology there, because I would argue that's more important than astrophysics for your life, right? And I have asked Columbia students what they think, and they tell me, oh, it's just so that we can have good conversations at cocktail parties.
Interesting.
But what is astrophysics going to do for your life? I mean, you can think of a lot of cool things. But, everybody's fascinated with space. That's why it's there. But microbiology and immunology can make you understand why you need to get vaccinated, for example.
Why the entire world shut down for,
So I don't know why it's not there. And they may argue, it's too complicated, you need other science. But you could make an immunology course that people could get,
You
right?
could make that course.
Yeah, and I think, that's what Sumali has demonstrated is that it is possible to agree on core concepts that you can get through.
Yes,
sure.
with immunology context. And especially true for community colleges. Community colleges don't have a standalone immunology course for the most part. Right. They have microbiology, but not immunology or introductory biology. So how do you seep in, sprinkle some of the immunology in there? That's one other way to do it. I think
part of it. I was just saying, I think part of it's also making sure that the immunology is accessible, because I think that in some cases that intro biology course is going to be taught by someone who's not an immunologist. And I think there's this idea out there of, immunology is very complicated. And since I'm not an immunologist, I can't go there because I can't understand it. Mm-hmm. And so making sure that we as a field are making it accessible so that it can go into the anatomy and physiology class, like you mentioned, or into the intro bio course or what have you. I think that was a big thing that we need to work on.
Yeah. This discussion is great. I was going to say we went the opposite direction. We had a junior level class, so like for the 3rd year in college, and we bumped it up to the senior level class. So it was a 3,000 level class. We bumped it to a 4,000 level class because we felt that it was too much. The students were overwhelmed. And so we figured, well, we'll just make it for only people who are really interested into it. And I thought we need this other course like you were talking about, Sumali, which is for non-majors, where you just kind of cover what's in the news and at a basic level, so that we're educating more broadly. Because getting this idea of getting immunology into other areas and not just people who want to go into medicine or veterinary medicine or graduate school in immunology. We got to go back.
I do it as a first year seminar on HIV.
Mm-hmm.
And so we can kind of talk about some social issues and things like that, but I'm also kind of sprinkling it in. And the fun part there is, those students are going to have the opportunity to take immunology later. So if I don't hit everything, I only hit the things I feel like hitting, it's okay.
So that's why I asked before about focused courses. So you could do an immunology of HIV/AIDS like you do, right? You could do an immunology of cancer. There are a whole bunch. Immunology and gene therapy, immunology and diagnostics. You could do all of those. They don't have to be huge courses, right? But you could do those as well.
Yep.
And small modules. So Immunoreach, going back to it, so the whole philosophy behind developing this research coordination network, it's really like a community of practice. And the tenet there was that the faculty who are immunologists like Phil and I, we have to pair up with people who are not teaching immunology generally and who are teaching introductory biology courses. So you're forced to think of, okay, what's the common language that we both can use to bring that into lower level courses? Maybe it's just a module that you're developing into evolutionary biology class. It doesn't have to be a standalone course per se. And hopefully with Immunoreach resources, that's what we're trying to accomplish, that make it accessible enough that it is not intimidating for a new instructor or a non-immunologist instructor to take those resources and incorporate them in the classes.
Right. I love the module concept because it can be smaller, digestible, but you can bring in a hot topic. That's great.
I'll just add that Michelle Pearson, I don't know that I mentioned Michelle's last name, at Spokane Falls Community College, and I have developed this module. It's under revision right now, but she happens to use the common learning management system known as Canvas. And in Canvas, you can share those modules as a shell where all the educational resources kind of are very easily imported. And we're looking for collaborators. We haven't disseminated this. Sumali was probably going to mention that all of these modules will eventually be published and open sourced, probably through the CUBES network. But in time, we'd love to have other community college instructors use this in their class, because we also want to collect data about how effective it is at helping students learn these concepts. And that's the educational research part of what we do, is we want to actually use assessment and collect data about whether it's working to penetrate and get those immunology concepts in the minds correctly of our students, regardless of what level they are.
So a call to action. I was going to ask for that. What are your calls to action? So if community college instructors, other instructors who are maybe non-immunologists, they should email either of you or Microbe TV, and then we can get them to you. What other calls to action would you like to put out there that if people that you hope people do or seek out? Immunoreach being one of them.
Yeah. So go ahead, Phil.
I was going to say so many things come to mind, but don't fear the immunology might be the call to action. That you've got some folks on screen today who are very happy to collaborate and work with you. We do want to bring those immunology examples and cases into other courses where we try and get rid of the jargon, use them for genetics, use them for science ethics, use them for public policy. I think that's a great way to think about not making immunology siloed and something only experts can comprehend or talk about. We really want to normalize it and push it out to a lot of different aspects of the science-related topics in our public dialogues. Go ahead, Sumali.
Yes, I would reiterate exactly what Phil said, but also adding that focusing on concepts is the key here. I remember a quote from Diane, who's at Michigan State University. She mentioned that if you don't assess what's important, whatever you're assessing will become important. So being mindful of your assessments and going back to those key concepts is super important, I think, including with immunology, even though we are very proud of our jargons and terms and receptors and cells.
And for those who are interested in teaching immunology, so we talked about this earlier in the episode, but it can be so for those who are already in graduate school, it can be challenging, particularly if you are not appointed to be a teaching assistant. That's not a part of your graduate program. You're at a medical school where your PI does not do that. That's a different scenario than maybe those who are in departments where you might be a teaching assistant. This would be maybe basic biology departments. I actually came from animal science. I was in an agricultural college, and teaching is very common in those colleges for graduate students. So what would you recommend for those that are in a position where they don't know where to look to get teaching experience? And then for those who are looking to go to grad school but want to focus on teaching, what would you say to those people?
So I can give an easy part to one of those answers. So one of the things that I have talked about with some of my students who have left Drew and have said, I want to be eventually a professor at a place like Drew, is I've mentioned that they might want to think about graduate schools at campuses that have some undergrads. Because there are some schools where you are at a campus where there aren't really any undergrads, and so you're less likely to get an opportunity to teach them. So that's one thing to maybe think about when thinking about graduate programs. I, as a graduate student, had to do some TAing, but then Harvard has an extension school which is basically like a night school, where I got extra TAing, where people who were coming back for education were doing that. And I also reached out to other universities in the Boston area to try to find places where I might be able to get some experience as well. So I think that thinking broadly could be really helpful, but I'm also really interested and thinking about things like those lunch seminars that I went to as a postdoc were great, but I also really would love to hear what other people think on this one.
Well, to just dovetail on what Brianne said, again, adding to your resume with some defined activities. So if you think about it in a sort of backwards design point, when you apply for that teaching job, you want to have an application that includes some teaching. But maybe that's, as you say, Steph, not part of what you're getting rewarded for as you're working on your own individual research project. So volunteer at your local science center, where you're going to be the smartest person in the room, right? And that's going to be very affirming. You're going to have to translate your immunology research to a 6-year-old asking about turtles. So the point would be that exercise, which could go on your resume as a volunteer activity, is very important. Sumali and I both benefited greatly from professional activities associated with our professional society, ASM. ASM has a bunch of webinars, a bunch of competencies that you could gain, whether it be diversity and inclusion, whether it be how to teach a laboratory course remotely. Again, even if it's just attending those webinars, you're using your free time to build a skill set. And that's not quite the same as teaching a course, which you may or may not have the opportunity to do, but volunteer and lean in, talk to people who are doing a lot of teaching, see if you could participate in some way to see behind the curtain or whatever you want to call it. And really, as Brianne said, know the language of education and prove that you could step into a position where you're in charge of a class right out of the box.
Yeah, so one way is that definitely, dealing with students is one part of our teaching, but also delivering content is another part. And I feel with today's day and age, it's easy to do that because we have free YouTube videos. So making your own portfolio with the content that you're passionate about, and how would you deliver it? It could be an animation, it could be something, a presentation that's interesting, or a puzzle worksheet that you've developed, or, whatever. That might be another way to build your portfolio as a teacher while in your graduate school.
I was actually going to suggest that because, in addition to the traditional methods, we have the internet now. And I agree, you could make your own course. It could be 1 or 2 YouTube videos. What I really like is live streaming, which I've come to do quite a bit. You can, and the reason that's cool is because there you have the class and you get used to answering questions that come from all over the place, right? And not just students, but people who aren't in school, people who are 95 years old, who have a different perspective on life. To get used to doing that, that'll really get you ready for teaching in a classroom. So I would say make videos. Yeah, that's great in whatever it is you're passionate about, but consider doing a live stream on a particular topic and hone your skills that way. It's very different.
Vincent mentioned that you made that transition in part because you were a good presenter, whether it's a scientific seminar. So again, building your communication skills, whether you're presenting your own work. Some schools have 3-minute thesis competitions. That's another place to demonstrate that you have the relational skills to interact with students, tell the story, and help students understand the story. And those might align more with someone who's in a very research-focused unit without some of those other opportunities.
And I was going to add to that, students are often reluctant to, give talks at, or put their name in the hat for giving talks at symposia or, local things or at national meetings. Just do it every time and take the experience is what I would say, because that's the only way you're going to get more comfortable and better at speaking and answering questions is just by doing it. So anytime you have a chance to do a journal club, do a journal club. Anytime you have a chance to give a talk, give a talk. Practice giving longer talks, shorter talks, and think creatively about transferable skills. Right? Because we often think, I don't have any experience in teaching, but you actually have quite a number of things that you're learning on the job as you're a graduate student and a postdoc that are quite transferable, that you may not have experience running a whole course with X number of students, but you certainly have experience standing in front of the lab and delivering information that you need to digest in a way that they can understand and field questions. And so those skills are quite transferable.
Or mentoring someone else in the lab in a project or a technique.
Yep, and answering the questions that inevitably come up. Working with undergrads, you do that a lot, right? They ask questions, you're like, "Oh, wow, I didn't think about that."
We were talking earlier about research versus teaching, and I would say that, a big part of my research is teaching because it's mentoring undergraduates about how to do research. So it's hard to separate.
Yeah, we use teaching research as a teaching tool.
Most labs will take on undergraduates if they're available as helpers, and that's a really great experience for the undergrad. But it's also building your mentoring teaching skills.
Exactly.
I'm interested in knowing how the pandemic has impacted your teaching approaches. Obviously the shift to only online is one thing, but what did you have to learn to do that, and how do you take those experiences back when you go in the classroom?
I'll go ahead and respond. And Sumali, I know you have. I'll let you formulate your response. The transition during the pandemic was challenging for me because when I get ready for a course and design it, and think about the activities or the case studies or how my learners will actually, get to the point of knowing and be able to then put out something that represents their knowledge on an assessment. I didn't have the luxury of that. My institution shifted at spring break. Students went home for spring break and never came back. Well, that meant that all the instructors spent their spring break trying to figure out what the heck was about to happen. One, ASM convened online meetings almost daily to try and have collaborators talk about it. And so again, I think we got better at sharing and collaborating and acknowledging that everybody was in the same boat. Some things translate well to the remote platform. Other things really don't. My lab class, which I sort of envisioned would be more of a demonstration-based learning experience, I learned that was not very effective because it was passive for my students. So I need my students to be doing more to really understand what was happening. And again, in the pandemic, we did things in a very rapid fashion, didn't have that kind of trial and error. We talked about 3 times to get it right. But if I had to do it over again, I would structure the lab and that sort of activity-based class very differently than I did in haste during the pandemic. Sumali, do you have an example?
Yeah, so I, same thing as you, Phil. We pivoted mid-semester of our spring 2020, and I was teaching immunology and with a lab, and we had a project for our local conference that they had to present at with the data that we were anticipating to collect. So everything kind of changed with the online mode. So one thing that, being creative at that point, having collaborations, I remember giving them the experience. If they can't do the technique, what can give them the experience, whether it's data analysis. So of course, having that data for them to analyze. Or for microbiology class, I remember giving them, we have to, do dilutions. Can you take juice and dilute that because that has color? So how does it, fade out? But a lot of different things. The one thing that for online courses that I learned for myself was I need to have feedback from the students. They need to tell me where they are. And for me, that's very interactive because that dictates my next module design. So for me, reflections became very important that yes, you're doing this, but how much of this is really understanding it? How are you applying it to your project, which is an upper level grant writing or a project writing? And having that way of communication. So Google Docs really helped going back and forth with the communication, having Padlets where they can converse with the classmates and me constantly. That was really important for me to help move forward. Otherwise, I felt like I'm losing my students on the online platform.
One thing I learned is that online students ask way more questions than in person. I mean, I get a lot of questions in my live courses, and then the first day of going back to in-person teaching, there were no questions. So I said, you guys need a Zoom chat to ask questions now?
Vincent, did you find that they asked more questions in the chat or did they unmute themselves and verbally ask?
It's a mixture. It was a mixture, right? A lot of people are not comfortable talking and
I guess that's what
yeah.
I was curious about is online gives them the option to type it and maybe they have anxiety about speaking.
No, I think that's probably correct, and I think that's a good way to accommodate. So I toyed with the idea of having a Zoom chat running while I'm lecturing. It was just another thing I couldn't handle, but I think I might do that.
That would be cool because then they could, even if they were there, right, if they're there, they could be on their phones and they could just type in the question to the chat.
And also the other thing is that often other students will answer the question if they know the answer. I really like that part of doing it. So
yeah, that peer learning is just really gold nuggets when it's student to student. It can be incorrect, but the fact is they're having that dialogue. Really great learning.
Well, that's what I like about the live stream. That the community, whoever's there, are having conversations among themselves. And I taught a live stream virology course last year, and I said, I love this because in class you can't do this. You can't be chatting with one another, but here you can. And you can get a lot done. It's really unique. Sorry, Brianne, go ahead.
I was just going to say, I think that I also, had to pivot mid-semester and had a lot of challenges with the teaching. But one thing I can say now is that the one benefit, there have been a lot of non-benefits, But the one benefit, I think, to teaching during a pandemic is the way that it has made me kind of be more creative. I think that there are maybe some ways where I was starting to get a little set in my ways about how I did certain things in class, and I couldn't do that anymore. I really had to sort of think about creative pedagogy, and I've kept that creativity going since then. And so that's the one nice part about it.
Can you give an example, Brian?
So one example is that I've changed how I kind of do some of my assignments because I need to have more frequent check-ins with my students. One of the things is that when we were doing our online labs, we made a lot of videos of us doing techniques. So we would sort of have a video and then have students doing data analysis or something and sort of have this where they were going back and forth between them. And a lot of my colleagues and I have kept the videos as part of our courses now. And so before the student has to come in and learn how to use a micropipette, we now assign them to watch the video from that 2020 about how to use a micropipette. So they have more idea of what they're doing. And so taking some of those resources and thinking about how to use them creatively has just been really useful, I think.
Okay, I'll add one more here because I've really loved it since the time I started using it. So in my classroom lecture setting, I used iClickers quite a bit, which is interactive. You have, information covered and you can get a reality check on how your students are taking that information. So, I miss that a lot with the online mode. We can add questions to Kaltura, but that's not the same. So I came across a software called H5P that can be integrated into your learning management system. And if you use WordPress, it can be freely installed as a plugin, and you can make resources using that, which allows to make it super interactive. So you can have a text and you can have multiple choice questions, true and false questions right along with it. You can add feedback so that if they get the answer wrong, they go back and correct that answer. They can read the feedback right away. And at the end, it's integrated into your learning management system. So you can see how many of those questions they got right and how did they perform, and they had that feedback right away. So it's a very formative way of doing it. But again, that kind of served as a substitute for my iClicker based interaction.
Yeah, I use Poll Everywhere. Which is a website that does a lot of similar things. As a student, I was kind of at the beginning of clickers in some of my classes, and I didn't like some of the ways that my professors use them. So for me, it's actually really important that I do my clicker stuff anonymously, so students don't feel like I'm judging them while they're still doing their learning. But I love Poll Everywhere as sort of anonymous clicker types of questions to help check in with students throughout class.
Yeah, I want to mention one more. I was just at a conference for a training that I was at, and they used Slido. And basically, it just put one of the QR codes on the screen. You just hold your phone up, and then it takes you right to the page where the question is. You answer the question. Slido.
S-L-I-D-E-O?
S-L-I-D-O. No S-L-I-D-O. S-L-I-D-O. Huh. And it worked really well. It was seamless and very real time. But yeah, Poll Everywhere is another phone-based thing. I mean, they can do it on the computer page, but they can use their phone and just text the answer.
So I use really Socrative, which I use in class, and you can also use it online as well. And it can also be anonymized. You can say no names required, and the students like that. And then at the end, you can put up a graph of everyone's answers and see how everyone did. And I like to do that periodically to see if they're forgetting things. And if not, we stop and re-explain it. So that was seamless because I used to use that in class, and then it was very easy to do on Zoom as well. So H5B, is that the name, Sumali?
H5P.
H5P. I searched for H5B and I got something different.
So does it have alternative interactive way? How do I say this? So Brianne, you're probably aware of it's like you can do word cloud, you can do answer,
with Poll Everywhere.
Choice.
That's why I like
you can even put a picture, and they can and
have them label
something or put a dot somewhere.
Cool.
So like you can have an antibody and say, where's the antigen binding site, and see where the dots are for each student and whether they all got them in the right place. So there's interesting things that different platforms can allow you to do, that it's not always just a multiple choice question or a true false question.
So H5P is super interesting. It has a plethora, which I've yet to explore all of them, but they have like crossword puzzles that you can make, or advent calendar that you can make, and fun things that, you can drag and drop here and insert pictures, or, do those kind of things, which makes it super interactive.
Got it. Okay. H5P.com. All right, cool.
We're not endorsing these for commercial use.
No, we're not advancing them that. But a lot of what we've talked about is how do we teach students in the classroom? But are there things that we can take that we've talked about that we can try to apply more broadly? Because I think that, the trust in science is being undermined, and getting, the thought process of science and some of the information, information is power, allaying fears about vaccines and so forth. Are there things that we can take that we've learned about exciting students and educating students in the classroom and apply that more broadly to society? And how might we do that?
So one of the things that I have started doing post-pandemic is really taking their curiosity about whatever claim they want to come up with, whether it's, let's say, a botanical source serves as antimicrobial, that's lavender oil, and letting them drive that project in terms of thinking, is that true? What's the evidence for that? Is that evidence coming from YouTube videos? Is that evidence coming from news? How strong is the evidence? Are there any systematic reviews on that? Are there any the data analysis done on that, meta-analysis? So thinking about different types of evidence and then working in context of their project, letting them evaluate the information, I think that has really, I have to think about ivermectin, which is, YouTube video is not enough for that, and things like that. So having a module, I have it very consciously incorporated into my teaching now.
So that's Immune episode 60. The show notes will be at microbe.tv/immune if you have any questions or comments, or if you have some experiences in education you'd like to share with us, immune@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Our guest today from Minnesota State University, Moorhead, Sumali Pandey. Thanks so much, Sumali.
Thank you for having me. It was a pleasure talking to all of you, really.
From Washington State University, Phil Mixter. Thanks a lot, Phil.
Great to be here. Honored to be among some of the superstars in science outreach. Thank you.
And okay if I share your Twitter handle, Phil?
Absolutely.
It's at Phil Mixter. All one
and Sumali,
word.
You have a Twitter as well, yes?
Yes, it's Microbial Pack.
Microbial Pack. Thank you. All right.
Pack. PATH. Short for pathogenesis.
All right, microbial path. Thank you. Cindy Leifer is at Cornell University, at Cindy Leifer on Twitter. Thanks, Cindy.
Thank you. I wish we had more time to talk, but we got to go teach. So,
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thanks. This was so much fun.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thank you so much. I really enjoyed this conversation, and I'm really sorry that we have to end it.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.